So the first question is add more P53. And then this one is, I think it's like determine if the GAD 45 and this one is decrease P53 levels. Okay, I think that's that one. Decreasing levels to see if it changes the activity of the GAD45 promoter and the UV damage to 293 T cells. Cool. All right, so starting with number 1. So you want to add more P53 protein. So this one, you're going to want to use a transfection overexpression plasmid. Transfection plasmid. And so that drawing is going to look like this. So it's circle 1. Go like that. Boom and boom. So this specific type uses a virus promoter. And then down here, you're going to want to put the gene of interest, and we're interested in P53. That goes there. And so this is, you do this, and then you use the transfection overexpression plasmid, add it to your cells, transfect, and then you can use a Western blot to verify that the P53 levels were increased. So a Western blot could look like that. So that could look like that. Yep. So for number 2, we're determining if the GAD45 promoter is active. So for this one, you don't use that type of plasmid. You're going to want to use a luciferase vector. So that drawing is going to look similar, but it's going to be a little different. So it's a circle again. One up there, one down there, and one down here. Loose goes down here. Then you put the promoter you're interested in, and we're interested in the GAD45 promoter. So that goes there. So for this one, when you use the luciferase vector, you're going to be measuring the activity of a promoter based on how much light is shown. And so if there's light, that means the promoter is active. But if there's no light or very little, that means it's either not inactive or just not really active. And so you could do an RLU. And then, so like light would show up here. and that would mean it's active, and then it can be either a little bit or just none at all. That would just mean it's inactive. So for number 3, it's decreasing p53 levels to see if it changes the activity of the GAD45 promoter in UV damaged 293 T cells. All right, so first you need to decrease the p53 levels. So you're going to want to use an siRNA, or you're going to do siRNA to lower p53 levels. And so that you're going to want to, so for this I would split it into either you could do 2 or 4 plates, and like one plate would be the control, which like you wouldn't do anything to, you just leave it alone. And then like the second plate you would do the siRNA to lower the p53 levels. So like control, and rate just like normal. And then for the other one you could do, well, this is like the manipulator one, so you can do, what are we doing here, lower p53. And then these, I would say that both of these plates would be UV damaged when it gets to that point. But so here you're going to do the siRNA to lower the p53 levels. You add to the cells, and then transfect. Transfect like 24 to 48 hours, roughly. And then you're going to want to add a GAD45 promoter luciferase plasmid. And here is when you expose the cells, like I'd say both plates, to UV light to damage them. So you expose it to UV light so you can get a response from the p53. So then you wait about like a day or so, or a few hours, just to let the damage set in, let the cells kind of react. And to see if the p53 levels actually did get lowered, use a Western blot. And then you can check the amount of light on the different plates. So compare control and I don't even know what the other plate is called. Compare the control and decreased p53 level plate. To see if the levels being decreased change the activity of the GAD25 or GAD45 promoter. And then, so I think the second part of that question was, how do you know if or how do you verify if the P53 levels were decreased? You'd use a Western blot. I think that's it. Cool.